Hello, welcome to another BYU-Idaho Tutoring Center video. This is about DNA transcription. Here's our DNA double-stranded, double helix, nitrogenous bases in between. Well, transcription is the process by which we go from our DNA, this is our DNA, gets transcribed to some RNA, usually it's a single-stranded RNA, and then from there we can translate to protein, but we're going to focus on this process going from DNA to RNA. And if what's it called? An analogy that I've often heard is that the DNA here is like it's going to test my art skills. It's like a textbook. This is our textbook. It's got all the information that is necessary for life, the genetic code, but we only want a small portion, so that's the DNA, and then transcription is like taking a little note card and copying maybe a paragraph from this book. So you can kind of think of it like that. The DNA is this huge molecule, and the transcription is the process of just taking a small snippet of the whole genome. And usually when we transcribe something, we transcribe a gene. So if here was a DNA molecule, and perhaps between here and here there's an actual gene that codes for protein. So now we know that RNA is being produced. We can understand the different types of RNA, the molecules that there are, and the different enzymes involved in transcribing the RNA molecules. In prokaryotes it's nice and simple, just one enzyme does everything. But in eukaryotes there are three different enzymes: RNA polymerase, polymerase 1, RNA polymerase 2, and RNA polymerase 3. And they all transcribe something slightly different. So polymerase 1 transcribes rRNA, and the R stands for ribosomal, the RNA that is inside a ribosome, and it transcribes all of those except for what's known as 5S rRNA. S stands for Svedberg units. It's a unit of basically how long, if you took this molecule, how long it would take for it to form back together after ultracentrifugation. Don't worry about it, you'll learn about it if you take further genetics classes. Just 5S, a little bit of a smaller RNA molecule, and that isn't transcribed by polymerase 1. All other ribosomal RNA is. And then polymerase 2, the most important one really, transcribes mRNA. Well, most important, never mind, they're all equally important. mRNA, the M stands for messenger RNA. This is our little note card. This is carrying the information that's going to be translated into a protein. And then polymerase 3 transcribes snRNA. This is small nuclear RNA. It's involved with SNRPs and the processing of pre-mRNA. Don't worry about that right now. You'll learn about it later on as you go on to RNA processing. Polymerase 3 also does the 5S rRNA. The one that's not transcribed here by polymerase 1 is transcribed by polymerase 3. And there are some other small minor RNA molecules that are transcribed by polymerase 3 that we just don't care about for this class. All you need to really understand is polymerase 1 transcribes rRNA, polymerase 2 transcribes mRNA, polymerase 3 transcribes small nuclear RNA, and the 5S rRNA. Just remember that this is the process in eukaryotes only. Prokaryotes, they just have one polymerase that transcribes all three of these different RNA molecules. So let's look here. If we have, let's just draw a quick DNA molecule. If here's the gene from maybe here to here, this is our gene. There is something before the gene known as a promoter. The promoter region is vital for transcription to occur. If we look at, zoom in on the promoter region, if here's our, I'll just draw one line. Right here is the beginning, the start point, or the first nucleotide in our gene, and we label this the +1 site, and then say here's the end of the gene, right, corresponding like here and here. A bunch of nitrogen spaces that need to be transcribed. This +1 is the initiation site. This is where transcription will begin, right here in this nitrogenous space. Going in this direction is known as upstream. I think. Yes, this direction is upstream, is towards the 3' end of the DNA, is upstream, and we count up from here in a negative fashion. So if this is a +1 site, the next nitrogenous space along, pretty sure it's a -1 site, and then -2, -3, -4, blah 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 blah. And if we were to continue on past here, the -10 site, there's a transcription, it's called the Tartar box, and that's located around this region. And this Tartar box is known as a consensus sequence, or promoter sequence motif. Basically, without this Tartar box, the RNA polymerase right here, maybe it's polymerase 2 because it's going to, RNA polymerase 2 is going to transcribe some mRNA. This is a gene that needs to be transcribed. The polymerase 2 will not bind to the promoter region without the Tartar box present, and about the -35 region further upstream, there is another consensus sequence that's known as the TT-GACA box, I guess. TT-GACA is the consensus sequence, meaning that there's a sequence that's either identical to this or similar around this the -35 upstream region, and then there's the Tartar box is usually TAAT or something like that. It's fairly standard across almost all prokaryotic species. This -35 sequence can vary slightly, but as long as there is something here, the -35 region and the -10 region, then the polymerase will bind. It will bind, it will attach to the RNA, to the DNA, it will attach to the DNA, and transcription will initiate. There are a few things that need to happen first. The process by which polymerase, RNA polymerase 2 attaches to the DNA is a slightly but not too complicated process. If here's the DNA, it's a 3' end, and we've got our +1 initiation site, and then terminal, this is the end of the gene, and it will continue on like that. And the -10, we've got the Tartar box, the -35 region, all upstream, we've got the TT-GACA, whatever. There's these two consensus sequences, and the distance between them is ideal for something known as sigma factor. Sigma factor is a part of polymerase 2. RNA polymerase 2 consists of 5 subunits. 5 subunits known as there's a alpha subunit, beta subunit, the sigma subunit, the beta prime subunit, and another alpha subunit. This first one, the sigma factor, the sigma subunit will bind, and then once that's attached, the other subunits will attach, and this entire structure, RNA polymerase 2, is otherwise known as the holoenzyme. So the sigma factor recognizes these 2 consensus sequences, and then that binds and it recruits the rest of the subunits to form the holoenzyme, which is RNA polymerase 2, and then transcription will begin. This protein complex, the holoenzyme, it has both helicase and polymerase abilities. Sorry, the pen's squeaky. I don't know if you heard that. Helicase means it can go, as it's transcribing, as it's going through and elongating the strand, it's unzipping it and then transcribing an RNA molecule. So as it reads, after a bit of time, the polymerase 2 is going to be here, and this red line is going to be our RNA molecule. So it's coming off here, and as it's reading each nitrogenous base, it's adding the complementary RNA nucleotide to this RNA strand, the messenger RNA, and then that will go out into the cytoplasm to be, it'll be processed, and once it, we're talking about prokaryotic. This is all happening in a prokaryote. This mRNA will be translated straight away into a protein in prokaryotes. Eukaryotes are a little bit different. The difference, everything I've spoken about so far has been prokaryotic. In eukaryotes, this -35 region can differ a little bit, and the tartar box usually in eukaryotes, they got the -20 tartar box instead, and the processes in the eukaryote in eukaryotic organisms are much more complicated, and you don't need to know too much about how eukaryotic transcription occurs. In BYU-Idaho, we're just going to focus on prokaryotic transcription. However, what you do need to know is, if here's our DNA and here's the promoter region, in eukaryotes there are a number of transcription factors, proteins that bind to specific regions in a specific order, and they're known as transcription factors. And usually transcription factor A through J, they will bind and recruit the polymerase 2 in order to begin the transcription process. And I made a mistake back here, sorry. The holoenzyme is not polymerase 2, it's just RNA polymerase. Sorry, in prokaryotes there's just one polymerase that does everything. I hope that helped. Thank you for watching.